We're going to continue on in chapter 14, which covers how eukaryotic cells sort and transmit chromosomes. This mini lecture will cover specifically the phases of the cell cycle. In our discussion, we'll talk about the 4 phases of the cell cycle, which include G1S, G2, and M, and we will also discuss the regulation of the cell cycle, and that is mediated via cyclins and cyclin-dependent kinases. When we say the cell cycle, what we mean is what is happening when a cell is in the process of dividing. And the cycle is going to include 4 key phases: G1S, G2, and M. G1 is the first growth phase, and this is simply where the cell is getting bigger. It's assessing what growth factors are around, what nutrients are available for it to undergo the energetically demanding process of mitosis. During S phase, this is DNA synthesis phase, this is where all of the genetic material is going to get replicated. And we talked through that process of DNA replication extensively already in chapter 9, and you know what's going on there with the involvement of DNA polymerase in replicating all of the genetic material. Following that process, we have the next key step, which is another growth phase known as G2. And following that second growth phase, we can then enter into mitosis, which is the process of the cell literally dividing and becoming 2 identical daughter cells relative to the initial parent cell that entered into the cell cycle. Now, a cell is not always dividing. It could be in a what we call G0 phase, in which it's not actively dividing. And so this is true for many of our cells that are not undergoing this process. So again, when we refer to DNA replication, that is going to all be happening in this S phase here, the synthesis phase. As we talk about what's physically happening to the chromosomes when the cell is actually dividing, I want to point out some key structural features. So again, we've already talked about the fact that you get 1 copy of every chromosome from mom and 1 from dad. So here potentially we're looking at a single chromosome that is coming from, say, mom. And after the S phase, it's going to look like the letter X. And the DNA here is identical. One sister chromatid is identical to the DNA in this sister chromatid. A couple of key things to keep in mind: the middle of that chromosome is known as the centromere. We already talked about the petite arm, the P arm. The Q arm is going to be that longer arm. At the tips of our chromosomes are known as our telomeres. And again, the DNA in the sister chromatids is identical to one another. This is what's getting replicated during DNA replication. Another key region to keep in mind is the kinetochore, which makes up this whole region around the centromere and is going to be involved in ultimately pulling these 2 identical sister chromatids apart during the process of mitosis. So when we look at what's actually happening in a cell during the process of mitosis, again, we've got the chromosome here from mom shown in red and the chromosome from dad shown in blue. After that synthesis phase, again, all the genetic material has been replicated, and the identical DNA is in this other sister chromatid, and it's connected here at the centromere, and around the centromere is the kinetochore. And this is the region to which those microtubules extending out from the centrosome is going to pull apart these 2 sister chromatids from one another. Another key term to keep in mind is the chromosome from mom and the chromosome from dad. These are known as our homologous chromosomes. And then again, the sister chromatids are identical to one another, which is the process of DNA replication. So when exactly would we want cell division to take place? Well, it's very important during reproduction. It's important during development when we go from that single fertilized egg to, in a matter of months, to a whole person. During tissue repair, cells will undergo rapid cell division as well. So there's key times where our cells need to be in the process of actively dividing. Your skin tissue, for example, is constantly regenerating itself, which is a result of cell division. There are other cells that do not actually divide, such as your human nerve cells. So if these get damaged, it's actually a permanent damage in which we cannot regenerate that nerve cell. There's other cells, such as those that comprise the liver, such as our hepatocytes, and those normally are not undergoing cell division, but if your liver undergoes major damage, then those cells can actually enter into a state of cell division and repair the liver. If we're talking about bacteria, it's a much simpler picture. So again, if we have our bacteria and our huge chromosome, that would be a big circle that we've already talked about. Prior to cell division, that entire genome is going to need to be replicated, and once it's replicated, these two cells will get pulled apart. We're going to pull those chromosomes apart, and then we are going to split those two cells, and this process is known as binary fission. We had an initial parental cell, and now we have two identical daughter cells to what had been that initial parental cell. One more time, just to clarify our terminology, shown here in purple we could say would be the chromosome from mom, and shown in green could be the chromosome from dad. And again, these are known as our homologous chromosomes, but following that S phase, the DNA replication phase, we are going to end up replicating this entire chromosome, the entire purple chromosome, and it will have this particular conformation, and these two sister chromatids are identical to one another. However, the homologous chromosomes are not identical to one another. What you got from dad versus what you got from mom is not identical. Okay, so now once again we are going to go through this process of the cell cycle, going from G1S, G2 to M. The G1 phase is sometimes referred to as the gap phase or the growth phase. S is going to stand for DNA synthesis or replication. G2 is that second gap, also known as a growth phase, and then M is going to be mitosis and cytokinesis. And cyto means cell, and this is literally the splitting of the cell into two cells. The G1S and G2 steps are known as interphase, and then once we enter into M, that's known as mitosis. And again, a cell does not have to be actively in the cell cycle. It could be removed out of the cell cycle, not actively dividing, which would be true for things like our nerve cells, in which case it's in a more dormant phase where it's not actively dividing, but the cell is surviving and expressing genes and has a particular function. It's just not in the process of active cell division. Okay, so again, during G0, this is known as our resting or normal phase, again, just not actively dividing. And for a cell to exit out of G0 and go into G1, that's going to require some signal from the cell that it's time to enter into that active growth phase. G1, which is the growth phase, this is going to be for a particular amount of time, and it's really where the cell is assessing if it has the appropriate nutrients in order to undergo this very energetically demanding process. During S phase, as we've already talked about, this is DNA replication, and that's where an exact replica of the genetic material takes place. We then enter into G2, in which case we're assessing whether or not all the appropriate replication took place before we enter into the mitosis phase, in which the cell will literally undergo separation of all those chromosomes and then splitting of the cell into two individual cells. All these phases, G1S and G2, again, is known as interphase, whereas mitosis it takes place for a much shorter amount of time, and ends up resulting into identical daughter cells. We have a couple of key checkpoints going between the G1 to S phase as well as between S and G2, where before the cell moves through the cell cycle, we try to make sure that everything is properly prepared, that the replication has taken place without any errors, such that prior to entering into mitosis, we make sure that everything is 100% as to the best of our ability before we take that single cell and make it into 2 cells. All right, so if we look here at G1, we've got 3 sets of chromosomes. The chromosomes from the mom, just for the sake of argument, let's say it's the red chromosomes, the chromosomes from dad are shown in blue. So that means we've got 3 chromosomes from mom, 3 chromosomes from dad. The longest chromosomes here, these would be our homologous chromosomes. Here we've got another homologous pair, and here's our medium ones. These would be our other homologous pair. So here the cell has entered into G1. Again, this is growth 1, and so we are assessing whether or not we have all the appropriate nutrients in order to undergo this process. The cell will then enter into the S phase. S again stands for DNA replication, so you'll see that each chromosome has now been replicated, but it's still connected here at the centromeres. And you can always identify your number of chromosomes by counting your centromeres. So we haven't doubled the amount of chromosomes, but we have doubled the amount of DNA. And so at this point, we're going to check and make sure that no errors were made. DNA polymerase has that proofreading capability that we talked about, and so we will have a lot of examination to make sure that what we are sending forward is properly replicated genetic material. The cell will then enter into that second phase of growth, in which again, we will be determining if we have enough nutrients, etc., for the cell to undergo the physical process of mitosis. Once that assessment has been made, the cell will then enter into mitosis, in which all those chromosomes are going to line up at the metaphase plate. And then by the end of mitosis, we will physically split the 2 cells in cytokinesis, such that we end up with 2 daughter cells that are looking exactly like the initial parental cell that entered into this cell cycle. Now, how is it that a cell knows it should go from that G0, that quiescent state, into the process of G1S, G2, and mitosis? Well, it's a result of 2 things. One is an external signal that's received by the cell in the form of growth factors, and these are also known as mitogens, and the name tells you what it's doing. It's generating mitosis. So mitogens are growth factors. The second way is by internal signals within the cell, which are known as cyclins. Here's an image from a figure taken from chapter 8, where we talked about cell signaling. And so here a mitogen would be the same thing as epidermal growth factor, shown here in this slide. An epidermal growth factor is initiating a signaling cascade that ultimately is going to lead to this cell undergoing cell division. And again, if you think about what is the epidermis, it's the skin. So this would be literally a skin cell that is going to undergo DNA replication and cell division, such that this cell will then become 2 daughter cells. So that would be an example of our external signals. Internal signals would be things like cyclins and the role of cyclin-dependent kinases. Cyclins are protein activator molecules that literally cycle through during the cell cycle. Cyclin-dependent kinases, on the other hand, are kinases. So you should immediately think, what is the role of a kinase? That is something that phosphorylates other protein targets. So this is going to cause a phosphorylation event. So cyclin-dependent kinases are proteins whose activities are going to be dependent on the cyclins. So here, what you can see is that the level of CDKs is constant. cyclin-dependent kinases are always around, but they are not going to get activated until we have a high enough level of our cyclin proteins. So as a cell progresses through the cell cycle, the level of your cyclins is going to cycle. So for instance, we've got cyclins that are specific to that G1 phase, and we're going to see that it's going to lead to an increase in protein expression of G1 cyclins. And once we reach a high enough level, that's going to lead to the activation of CDKs. Once those CDKs are activated, that's going to lead to phosphorylation. And so we have a whole series of cell cycle specific proteins that are going up and down as it progresses through the cell cycle. Again, those cyclins are going to be produced in response to growth factors, those external cell signals, and those concentrations are going to increase over time in response to stimulation by those growth factors. And again, CDK activity is only going to be fully activated when we have a high enough concentration of the cyclins at specific points within the cell cycle. And activation of that CDK is basically going to push the cell through the cell cycle so that it can transition from G1 to S or from S to G2, etc. Here is showing the level of expression of a series of cyclin-dependent proteins. So here's a G1 cyclin, and you can see it increases during G1 and then kind of plateaus and eventually will drastically decrease. This cyclin, this G1S cyclin, is critical to push the cell through going from G1 to S, and you can see there's a stark increase just prior to the S phase, and then we're going to see a rapid decline. And again, once these cyclins reach a high enough concentration, that's going to activate the cyclin-dependent kinases, which will then phosphorylate key target proteins. Okay, here's yet one more way of looking at this. So if, for instance, we've got a particular cyclin that is going to bind and activate the cyclin-dependent kinase, once that concentration is high enough, we're going to then be able to push the cell through from G1 to S. The cyclin is going to degrade, whereas the CDK is just going to consistently be around. We then have a different cyclin that's going to help push the cell through from G2 to M towards mitosis. Once that concentration, that protein concentration is high enough, it will likewise activate CDK. CDK can then help the cell move through into that next critical phase, in this particular instance into mitosis, and we likewise will see the degradation of this particular cyclin such that the cell won't continually be going through mitosis. We just want to push it through to that next phase. Okay. So again, we've got these key checkpoints throughout the cell cycle. In G1, we've got a particular checkpoint that's trying to assess if we've got appropriate growth factors, enough space for the cell to actually divide. Is any DNA damaged at this point? If it is, we don't want to be dividing a damaged cell, and we would actually drive that cell into apoptosis or cell death. Another key checkpoint is going to come at the G2 to M transition. Here again, we've just replicated all the DNA in that S phase. The cell is now grown and we're about to divide. So we want to make sure, is all the DNA appropriately replicated? And is this cell really ready to become two new daughter cells? Another key important checkpoint is in mitosis. When all the chromosomes are lined up at the metaphase plate, we'll be checking there as well whether or not this cell should progress through and complete the process of mitosis and ultimately cytokinesis. All right, so again, we've got these three critical checkpoints in which we want to make sure that the cell that we are asking to divide and become two is 100% accurate, and it's got all the resources it needs to undergo this particular